Hello, this is Miss Dagenford, and I hope that, sorry about that, I hope that we can hit the ground running tomorrow with this video. We started today by looking at transcription and an overall level and the code for building proteins. So we're going to look a little bit more in detail of transcription. We're going to compare both eukaryotic and prokaryotic. So we're going to start with prokaryotic. Now what you're going to learn, what you're going to see over all of the things that we look at with gene regulation and modification and gene and development of the cells, that everything with eukaryotic is more complicated. And so the prokaryotic oftentimes is a few steps, boom, we got it done, yay. And then the eukaryotic, it's always more complicated. But it does usually follow a similar basic step. So let's go ahead and get started with the prokaryotic. Now, a quick review. We have our DNA double helix here. So what you're seeing, these are the two strands of the DNA. So this is our double helix. They're just not showing the spirals. And here we have our 5' end. So you should be thinking that this end should be, and that makes this end what? And then therefore this end is. Now, and you might have seen the picture below, but if that's 5', then this one over here is 3'. Here's 3' on the other strand, and then this makes it 5'. Now, in order to open up our DNA double helix, we have an enzyme, RNA polymerase, and it's going to come along to a specific region. And this region is known as the promoter because it promotes. You can think about that. And it opens up that promoter, that double helix. And what it's going to do is start transcribing the strand, and this one is, it's going to be called the template strand. And that template strand, think about in our class, the template is what you use to make something. Well, the template strand is what we use to make our mRNA. And so it's going to land on the promoter region, and it's going to start unzipping. And at that point, it's just going to start connecting our RNA nucleotides. And think about what this guy is going to do. It's going to travel down. So if this is like right here in a few seconds or milliseconds, it's going to be down here, and it's going to be even longer. So it's going from the left to the right. Now, because it's going to the right, anything to the left is called upstream. And you can think about the way that the river flows. If the river is flowing this way, then upstream is up from where it's currently now. It's going up the stream as opposed to downstream. So we use that language for our transcription. So here's our mRNA strand, and just like DNA, you can only add to the 3' end. So our RNA polymerase is adding to this 3' end as it travels. And so just like DNA replication, you have to add to that end. So we go from 5' to 3'. And so we're going to be making our mRNA strand from that one. Now, in prokaryotic transcription, it's pretty easy. It grabs onto the promoter, it happily unzips the DNA for about 10 nucleotides at a time, and it adds to the growing strand the right nucleotide. It connects the backbone, and it just happily goes on. It usually hits a terminator region, which loosens it up. There are some pretty cool elaborations to this, like how does it cause it to loosen and work free, but the basics are there. That's it lands, it transcribes, and then it comes off. Now, if this is a really high demand transcript, you might have another RNA polymerase right behind it, landing on the promoter and then making another transcript and then making another transcript. So we can make multiple mRNA transcripts. Now, if we see eukaryotic, and remember, it's just going to be more complicated, eukaryotic, it does have a promoter region, but it's just a little bit more complicated. This promoter region in eukaryotic cells, it's very consistent. We see a region where we have a lot of A's and T's in our DNA strand. And so much so that they named it the Tata box. I know that's a weird name, but you understand why they do it. There's our Tata box right there. And it's going to be where our RNA polymerase lands. So we have this promoter region. And the other thing we notice here are the transcription factors. Now, we don't see those in prokaryotic, so this is specific to eukaryotic cells. So transcription factors. And you want to think about the fact that these are similar to an on-off switch because it's often needed to get the attention of our RNA polymerase. I often liken it to if the prokaryotic system, if the promoter is like the landing pad, the helicopter landing pad, like land here, then the eukaryotic is our landing pad with our circle for our helicopter with lights shining around it saying land here, and the lights are directing the helicopter to land. But not only that, you've got the little guys have the little wands that are saying, here, not only do you need the lights and the pad, but we're going to direct you down there, and then throw down a loudspeaker for good effect. And so you kind of have this very complicated system. All of these things contribute to this go signal, this land here. Now, this oftentimes allows us to now regulate because these transcription factors are often different for the type of the cell. So for instance, if you have a stomach cell, the stomach cell is going to have like transcription factors A, B, and C. A nerve cell might have transcription factors D, E, and F. And then our skin cells might have transcription factors G, H, and I. And then they all might have some things in common. There are housekeeping genes. So they might all have like L and M on. But these are going to be the genes that are turned on. And then for each one, if they're a stomach cell, they don't need the genes for the nerve cell on, so they would be off. And these transcription factors are a big key to this because if this is A and B, then we know this is going to be a gene for a stomach cell. Now, one thing I want you to ponder is the fact that what if these transcription factors, which are proteins in and of themselves, most of them, if there's a mutation in the transcription factors? So what if there is a mutation in the transcription factor? And I want you to ponder that and see what we come up with. And I hope you could see that if we don't have A right there, then that might be a crucial problem with turning that gene on. And you might get a glimpse of how all of these things might be regulated. Now, one thing I did forget about, and that was the promoter, the A and T region. There is a reason for that. Remember, A's and T's, they only have 2 hydrogen bonds between them. So if you have only A and T in that region, this becomes very loose, or at least easier to open up than if you had a lot of G's and C's, which have 3 hydrogen bonds between them. And then if we look down here with all of our transcription factors landing and saying, okay, RNA polymerase, start here. So there we have our ta-ta box right here. Here we have the rest of the promoter region, and now it's going to be the same thing. It's going to open up our DNA strand, and it's going to start just as the prokaryotic ones are. Now we've got some similarities. Now, in detail, the elongation is actually pretty simple. Once it starts, it happily transcribes, opening it up for about 10 nucleotides, and then notice it's just going to match it, A to T, A. Now, here's the tricky one. Remember, instead of a T, it needs to be a U, a uracil. G to C, G to C, T to A, and it's just going to add it on, and once it matches, it's going to catalyze that bond between the backbone, the phosphate, and in this case, the ribose. And then once it gets to the end, there's usually a termination sequence. And in eukaryotic cells, it's called a polyadenylation or poly-A. And this time you get lots and lots and lots of A's and U's. In fact, up to like 10 to 35 nucleotides where it's just kind of loosened up, and then it usually can fall off at that point. And again, just as in prokaryotes, if this is a DNA that's in high demand, you could have consistent transcription happening on this high demand DNA gene. Now, the modification does play a huge role because in eukaryotic cells, so let's emphasize that that's only in eukaryotic cells, you're going to modify this mRNA. You have the 5' over here, and you're going to add a very unique 5' guanine cap to this. And we're going to add that there. And then we have the protein coding regions, some untranslated regions, the UTR untranslated regions. And then we're going to add a 3' poly A tail. And this could go on hundreds of bases long. Then the big thing, though, is we have these regions that can be snipped out or spliced out. And these areas of that can be snipped out are called introns. So the introns, what you want to think about is the introns stay inside, introns inside. The exons exit the nucleus. So you snip these out, you have to cut them out, and then you get a mature mRNA. Now, one thing I really want to drive home is the fact that this is a mature mRNA for a eukaryotic cell. If we wanted to, we could take this mature mRNA. This is the one that we could use for if we want to add it to a bacteria. Now, this is a transient molecule. It will be broken down. So what we often will do is we'll actually use an enzyme called reverse transcriptase to go back and make a copy of DNA, and that's called cDNA. And that allows us to now use that copy of DNA to put it into a bacteria to make whatever we want to make. The best example is insulin, where we can use a bacteria to make our insulin. So real quickly, and we'll talk about that tomorrow as well. So how do these SNPs occur? Well, they occur by the use of another enzyme, and it's a big consortium of nucleic acids and proteins. So we call them SNRPs. And they're called SNRPs because, well, the name is kind of blended to make it sound like it. It's small nuclear ribonuclear proteins, and they're made up of RNA and protein. And you put them together in this big consortium, and we get a spliceosome. And notice particularly the spliceosome has regions of open nucleic acids that can bind hydrogen bond to the RNA here and then here and pull the two ends together while the enzyme snips them and then puts them together to make the mature RNA. So we'll work on that and practice that tomorrow, but this is a good beginning for the details. Have a nice night.